In this video, we'll talk about pancreatic beta cell function. Beta cells import sugar at a rate that is proportional to blood sugar. They secrete insulin at a rate that is proportional to blood sugar. Of course, the more insulin they make, the more they are working to reduce blood sugar. So this is essentially the key to the negative feedback loop that keeps blood sugar from being too high. So as blood sugar increases, blood sugar is imported at an increasing rate. So as blood sugar increases, the rate of import increases over most of the physiological range of blood sugars. This purple range of blood sugar is the fasting range. So that's the homeostatic level of blood sugar, essentially. That's the goal, the target amount of sugar that's going to be in the blood. When you eat a big meal, blood sugar goes up. When you eat a big meal, blood sugar goes up and more blood sugar enters these beta cells. Typically, cells import sugar and then turn it into glucose and then turn it into glucose phosphate using an enzyme called hexokinase. And hexokinase has kinetics that are very steep. It's got a affinity for glucose that's very high. And so throughout the range of sugar concentrations that occur in the body, the physiological range, the quantity of sugar that's available for this enzyme keeps it running at Vmax. This is not true in a beta cell. A beta cell has glucokinase, and glucokinase has an affinity for glucose that enables it to make more and more glucose phosphate as a function of how much sugar it sees. Notice from this process that glucose is not going to be maintained in these cells within some homeostatic range. Instead, it's going to change. So you can't use the Km of this enzyme to figure out how much substrate is present because the substrate level for glucokinase is always changing. It's changing because the import rate changes with changes in blood sugar. So the more blood sugar there is, the more sugar comes in, and the faster glucokinase works, unlike some other kinds of cells. So glucokinase is the red graph there. Glucose is processed into glucose phosphate in a way that increases over a big range of blood sugars. The more glucose phosphate you make, the faster glycolysis works. Notice that PFK is not the rate-limiting step in this process. PFK does not determine how much metabolism goes on. It does not determine, only in these cells now, how much ATP is made per unit time. Okay, so essentially the activity of glucokinase dictates the rate of ATP production. Here is the sodium-potassium pump. It's doing its normal thing. I'm not going to draw details because, boy, by now you just have to know this stuff. So because of the activity of that sodium-potassium pump and the leak channels that probably are around, there is a typical membrane potential of about -70 in one of these cells. Up here would be 0. But when ATP levels increase, so the more blood sugar there is, the faster glycolysis works, the more ATP is present. When ATP levels build up, that takes a normally open potassium channel and closes it. Hopefully you realize that potassium at resting potential leaves the cell if a channel is open. Now the channel isn't open, so now that potassium isn't leaving. So if potassium was leaving in these cells, and these cells were maintaining a minus 70 membrane potential, resting potential, now that blood sugar has increased the quantity of ATP in these cells, they are no longer going to be at minus 70. They are going to depolarize. So this process, this closure, is depolarizing the membrane. Depolarization of a beta cell membrane opens a voltage-gated channel. The channel I've drawn over here in purple, I can't find my pen, here it is a calcium channel. So calcium comes in, voltage-gated channel. Calcium comes in. Calcium signals insulin-containing vesicles to exocytose. So they do. When they do, insulin enters the interstitial fluid and then diffuses to the blood. This is the response to increases in blood glucose levels. And whenever you talk about blood sugar, you're talking about blood glucose. But these days especially, we ate another sugar in pretty large quantities, and that sugar is called fructose. Now, fructose isn't, something to be too ashamed of eating, since every time you eat fruit, you eat lots of fructose. But fructose doesn't enter beta cells. Fructose instead is detected by beta cells because they have a receptor, very much like the sweet taste receptor on your tongue. And that fructose doesn't do anything unless calcium is present in these cells. But if calcium is present, if calcium is interacting with this channel here, the TRPM channel, if you get both calcium in the cell and fructose attached to the cell at this receptor, you get this TRPM sodium channel opening. And when that opens, in comes some sodium. And what is that going to do? That is going to depolarize the cell as well. So it's doing the same thing, potassium not leaving the cell. So this is the effect of having calcium kept in. And this is the effect of potassium kept in and sodium let in, so that you get a more dramatic depolarization, or a greater extent of depolarization. Greater extent of depolarization keeps these channels open longer, and this calcium coming in for longer, and so more insulin is secreted out into the interstitial fluid and enters the blood.